Hello, everyone. In previous videos, we've talked about how we can use ANOVA to assess whether or not there are differences in means among multiple groups. And when we do this, we test the single null hypothesis that all of the means are equal to one another. And when we reject this null hypothesis, then we know that there are differences among means somewhere in the data, but we don't know where those mean differences actually are. So what we're going to talk about in this video is how we can go one step further and identify which means are actually different from which other ones. If you remember, in one of the previous videos, we talked about this example from Turco et al., where they were studying how the gill morphology of mango rivulus is affected by the fish being exposed to air and air breathing. This is an amphibious fish that spends time in the water, but can also breathe air outside of the water. And they do this by packing their gill lamellae with these things called interlamellar cell masses that help to keep the lamellae open and exchanging oxygen. So Turco et al. Exposed rivulus fish to different treatments, either control, immersion, or recovery scenarios, and measured the interlamellar cell masses, or how packed in the gills were with these interlamellar cell masses in these three different treatments. And you can see from the conclusion of their paper that the mean interlamellar cell mass height differed significantly between the three subgroups. But which of these groups are different from which? Our ANOVA doesn't actually tell us this. So what should we do? Well, we could go through and do pairwise comparisons, comparing control treatments to immersion treatments, control to recovery, or immersion to recovery. But in this case, we're performing three separate t-tests. And as we talked about before, as our motivation for performing our ANOVA, when we do these pairwise comparisons, we will inflate our experiment-wise error rate. So if each comparison has a comparison-wide error rate, or alpha, of 0.05, then our experiment-wide error rate is going to be much bigger than 0.05. In this case, where we've got three groups, it's not going to be a lot bigger, but in other cases where we've got lots of groups that we're comparing, it could be much, much bigger. So what should we do? Well, in previous videos, we've already talked about how we might use a Bonferroni correction to adjust our alpha level to compensate for the fact that we're performing multiple tests of a single biological hypothesis. So in this case, we could perform a Bonferroni correction where we perform t-tests between each combination of groups, but instead of testing each of these comparisons at an alpha of alpha, or 0.05, we divide this alpha by the number of tests that we're going to perform. So in this case of three groups, we're going to be performing three separate pairwise tests, and each of those could be done at an adjusted alpha level of 0.017. But it turns out that just like before, this Bonferroni correction method is actually a little bit overly conservative. It corrects too much. So there are other post hoc methods that we can use. And what I mean by a post hoc method is an after the fact method. So we perform our ANOVA first to determine whether or not there are differences among groups. And then we perform these post hoc pairwise comparisons to see where those differences are. Now, we had talked previously about questionable research practices and the idea of performing post hoc analyses or generating hypotheses post hoc was a questionable research practice. In this case, these post hoc analyses are not questionable. This is how you actually determine where those differences are after you've found a significant difference in your ANOVA. So we have lots of different post hoc tests that have been developed that are better than the Bonferroni correction, but there are a lot of them out there. One of them called the Fisher least significant difference test, another one called the Tukey's honest significant difference test. There's lots out there. They're pretty interchangeable. I tend to use the Tukey test for the work that I do, but other people will use others for other purposes. There are differences between them, and if we were to get really technical, there might be some situations where one is better than the other, but for the most part, they're all pretty good. So if you want a good one to use, then maybe the Tukey test is a good one. That's the one, as I said, that I use. So let's walk through an example then of how this might actually work. Singaravelan et al. In 2005 did a study where they gave bees the choice of foraging between artificial flowers that controlled pure sucrose or sucrose spiked with caffeine to see if caffeine acted as a deterrent to foraging by bees. And they performed their analysis with 4 different treatment groups and determined that there were significant differences among these groups from their ANOVA, where the p-value from the ANOVA was less than 0.05. But where do these differences lie among these different treatment groups that correspond to different caffeine concentrations in these artificial flowers? So let's use a Tukey honest significant difference test to determine which means are different from which other means. So this pairwise test then makes sure that the probability of making at least one type 1 error remains no greater than our significance level of 0.05. So this test is going to adjust the alpha level for each of our pairwise tests to keep that experiment-wide error rate below 0.05. So we can specify our ANOVA model here using the AOV command in R, and we're going to save that as an object called DF. And we can then use this Tukey HSD command to perform a Tukey's honest significance difference test of this ANOVA model DF. And when we do this, the results look something like this for this particular data set, where you can see that the p-values, the adjusted p-values, are indicated in the right-hand column, corresponding to each of the pairwise comparisons that were made in this analysis. So in our comparison of 4 different groups, then we have 6 different pairwise comparisons, and 2 of these pairwise comparisons had p-values less than 0.05. Now, it's important to note that these p-values that are reported in the right-hand column are already adjusted by the Tukey test. So we don't need to do any further adjustments to them. They've already been corrected for the fact that we have performed multiple pairwise tests. And so from this analysis, then, we can conclude that first of all, our ANOVA identified that there were differences among groups, and second, that our Tukey post hoc test identified that the 100 parts per million treatment was different from the 150 parts per million treatment, and the 100 parts per million treatment was also different from the 200 parts per million treatment. So we can indicate this on our figure by including letters on the figure. And the tradition in science is to use the same letter to represent means that are not significantly different from one another. So for example, we have the letter A for these 2 treatments here to indicate that the 50 parts per million treatment and the 100 parts per million treatment are not different from one another. Furthermore, we also have the letters B up here for the 50, 150, and 200 part per million treatment to indicate that these 3 groups are not different from one another. So what we can infer from this figure then is that means that have different letters are different from one another. So we can see that the 100 parts per million treatment has only the letter A, and these 2 treatments have only the letter B. So we know that the 100 parts per million treatment was significantly lower than the 150, and the 100 parts per million treatment was also significantly lower than the 200 parts per million treatment. So Turco et al. Also performed a Tukey post hoc test and reported the results in their paper like this. The interlamellar cell mass was significantly enlarged in the recovery subgroup relative to both the control subgroup and immersion subgroup. There was no difference in interlamellar cell mass height between the control and immersion subgroups. Okay, and we can tell that there was no difference between the control and the immersion subgroups because those two treatments share the same letter. Whereas the recovery treatment was significantly higher than both of these other treatments because it has a unique letter. So how do we go about assigning letters to treatments? In these cases, it's relatively straightforward, but let me tell you, it can get pretty complicated pretty fast. The way to do this is to follow a series of steps. When assigning letters to treatment means, first of all, arrange the groups in order of increasing or decreasing means. Second, use our post hoc statistics to make comparisons among these means. And finally, draw lines connecting those means that aren't different and assign letters to the means that share the same line. So let's walk through an example of this. Here we have an ANOVA with 4 treatment groups corresponding to oyster density that was either absent, low, intermediate, or high, where our response variable is the below ground biomass. So here I've arranged these treatment groups in decreasing order of their means, from highest to lowest. Now, let's say we performed an ANOVA that determined that there were, in fact, differences among these means. How can we assign letters to represent where these differences lie? Okay, well, first of all, here are our steps again to arrange the groups in order of decreasing means, which I've already done here in both the figure and in the data. And then let's say we use a post hoc test to determine which means are different from which other means. And we're then going to draw some lines to connect to the means that aren't different. Okay, so in this example, the none treatment is significantly different from the high treatment in our post hoc test. Now, because these are different from one another, we don't draw any lines connecting anything. But in our post hoc test, the adjusted p-value indicated that the none treatment was not significantly different from the intermediate treatment. So what we do then is to draw a line spanning between none and intermediate. Now, because we've ordered these in decreasing order, because none is not different from intermediate, then none is also not different from low, and low is not different from intermediate. So we don't need to make these additional comparisons. We know that all 3 of these means are not different from one another. Okay, so let's next move on to the other end and compare high to low. Okay, and we find that in this case, high and low are different from one another, so we don't draw anything. But high and intermediate are not different from one another. And because they're not different from one another, we draw a line connecting high and intermediate. So high and low were significantly different, so we do nothing. High and intermediate were not significantly different from one another. So we draw a line connecting the 2 of those together. Okay, and now we've made all of the possible comparisons. We know that these 3 are not different from one another, and these 2 are not different from one another. And so how do we assign our letters? Okay, well, we can see that the only line that corresponds to none is our A line, so we assign the letter A to none and low. Intermediate is shared by both the A line and the B line, so we assign the letters A and B to the intermediate treatment. And the high treatment has only the B line associated with it. So we assign only the letter B to the high treatment. So this is a way that works in all circumstances to assign letters to treatments. And as I mentioned before, things can get really quite complicated depending on the patterns of what is significant from what. So it can get really confusing assigning letters to our treatment means. So I'd suggest you follow these simple rules as a way to keep track of which means are different from which other means. So while an ANOVA can tell us whether there are differences among means, it doesn't tell us which ones are different from which other ones. So instead, we use post hoc tests after an ANOVA to make comparisons between specific means while correcting for these multiple tests. Now, it's important to note that if our ANOVA is not significant, then there's no point in doing a post hoc test. All of the post hoc comparisons will be non-significant when our ANOVA is non-significant. Now, there is this very weird situation that can occur sometimes where our ANOVA is significant, indicating that there are differences among groups, but our post hoc test doesn't identify any pairwise significance. And this occurs because of those adjusted alpha levels, that it's possible for there to be enough variation among groups for those differences among groups to be significant, but no single pairs of groups are different enough for those pairwise differences to be significant. Finally, we can graphically depict these differences among means by using letters to indicate which group means are different from which other group means.